earlier session of immunological part. Earlier we saw in that immunology, what is immunology, why it is important, and then the two type of immunological factors, that is innate immune system and cellular acquired immune system. That innate immune system itself, a lot of categories are there. That one is that different factors, that is general factor, cellular factor and humoral factor. The last session we look into that, the general factors, that is three types. You can refer with the earlier session video. And then today we are going to discuss about the cellular factor. The cellular factor itself, we have the 5 different type of factors. It is important to the following the cellular factors. One is the microbiota, second one is the natural killing cells, NK cells, the phagocytosis, inflammatory responses and toll-like receptors. In this, the last session we looked about toll-like receptors, how it is work and then why it is important in terms of immune responses. Today, what we are going to be look into that another 2 important characters of cellular factors. That one is the phagocytosis, another one will be the inflammatory responses. That phagocytosis, what is phagocytosis? We know that W.B.C, that is the white blood cells in our body, we have different type of cells. Among that cells, we have the 5 different cells, that is the macrophage, monocyte and neutrophil, leukocytes and then other cells. In these, the macrophage, that is the major bigger cell which is actually working as a phagocyte cells. This phagocytosis cells, it is very important. It will be work on that immune responses, particularly on the engulfing methodology. So we will go into the next slides, how it is work and then why it is important in the immune responses. So here I saw that phagocytosis reaction. I saw that one cell with the nucleus and then another one will be the microorganism which is actually causing the damages to the cells. If any cell is responding, particularly on macrophage, it is responding to that the particular microorganisms, how it will be respond. So for this phagocytosis action, we know that we have the immune cells, particularly on neutrophil, macrophage, mast cells and dendritic cells. All these cells are known to be professional phagocytes. Why? Because this is the professional antigen representing cells or presenting cells. So these cells are actively working into that particular immune responses. So if we see in that, in the phagocytosis itself, we have the 5 different steps involved to complete that particular the microorganism infection. The step 1 is the binding, step 2 is the engulfment, step 3 is the phagosome, step 4 will be the lysosome fusion, and then step 5 will be the antigen presentation. If you look into that, each and every step, how it is involved and then why it is important. The step 1 is a binding. It is known to be the non-specific cell surface receptors. So in these binding steps, if you see the particular cell with the nucleus, it is try to make that understanding or try to make that link with the external body, that is a foreign body, that may be a virus, that may be a bacteria, that may be other group of parasites. If first what the phagocytosis action will do in the first step itself, the bind with that the particular foreign body. For this, it will use that the non-specific cell surface receptors. So that receptors is known to be a TLR, MR and CD14. If you look at that, these cell surface will create these receptors to make the connection with the foreign body. If the foreign body is accepted that, and then it will be started to produce that indirect opsonization method to produce C3b. In this C3b, and then these particular non-specific cell surface receptors will work together and then form that the linkage between the cell surface and the foreign body. In this process may be happen in any pathway which is responsible for the immune responses pathways, particularly on classical pathway, alternate pathway, or MBL pathway, monos binding lactin pathway. Any one of the pathway involved, and then it will be start to bind between that cell and that particular foreign organism. Then what it will do, that may be using the indirect opsonization using C3b, or direct opsonization with that FC receptors with that particular antigen or antibody interactions. So we will look into this part in that next sessions or maybe. So then the first step will be over, then the second step will start in the process of engulfment. We all know that how the pseudopods work in the amoeba, so protozoans. So in the same way, this step 2 engulfment is also will work. First the prey will be, the particular foreign body will be intact using the pseudopods, and then it will be covered immediately. Then it make that one cell wall towards that the particular foreign materials. So then the step 2 engulfment is over, then the step 3, that is the very important step, that is known to be a phagosome step. This step 3 will be that what it will do after the engulfment, the foreign body is covered by one cell wall. That is the temporary cell wall. Why it is created? Because this phagosome reaction or response is mainly depending on two types. One is the oxygen dependent type, ODD, or OID, oxygen independent digestion. So what it will do, once that foreign body enter to the macrophage cell, then it will be form the temporary cell wall. That cell wall itself, what it will be do, oxygen dependent method, first we will go one by one. Oxygen dependent method itself, it tried to reduce the oxygen concentration within that the temporary cell wall. Then automatically the problem will start the respiration, then automatically the particular foreign material or body will be direct to death. If otherwise, it also will do that oxygen independent digestion. In this method, what it will do, it will try to make that the oxygen is converted to OH and H2O, then it will be converted towards the nitric acid. So nitric acid, once it is the gaseous form, then automatically the particular foreign body try to death in that stage itself. If once in this stage the microorganism or the foreign body which is entered into that macrophage cell, then immediately after that thing, the foreign body will not kept in that cell for a long time, then the step 3 is followed by step 4 will stop, that is that lysosome fusion. Once the lysosomes are produced from Golgi complex, then the lysosome will be entered to that particular cell, the temporary cell wall, which is created in the step 2. Then the lysosome, the granules of the lysosome, then it will be fused with that particular dead particles, then it will be digested, and then entirely it will be known to be the residues of that particular material or foreign body. If that particular foreign body, that may be happen in the different type, that may be a lysosome fusion, that may be enzymes like peptolytic, hydrolytics, or antimicrobial peptides, or acidic reactions like OPHL and something, or lactoferrin concentration will be increased, then the residues will be kept in that cell wall. Once the residues are prepared in this step 4, then the step 5 will be the antigen presentation. if that residues are taken out from that inside the cell wall, cell macrophage cell to outside through that major histocomponent complex 2, in that pathway itself, it will be so into that antigen representation. That antigen is known to be that the residues. So, because based on these all 5 steps, we know that phagocytosis is not at a simple job, but it will be happen in our body within a few minutes, and then it will be try to avoid that foreign body enter to our body to make the problems. So, for these, what are our other enzymes or the reaction responses will be happen, particularly in these action that inflammation will be happen. From that inflammation itself, we have that different type of interleukins like 1, 6, 12 and then TNF-A and even interleukin 8. So we will go that particular interleukin where it is coming from. That interleukins are primarily known to be the pro-inflammatory cytokines. That may be in interleukin 8 will be a chemotoxics for leukocytes, and then interleukin 1 is the activate T lymphocyte and macrophages, and interleukin 6 is the activate T and B lymphocyte. TNF-A is known to be that activate endothelium cells, and then IL-15 interleukin 12 is known to be active natural killing cells. Once all the interleukins will work when the active macrophage was activated after the infection of the microbial contamination. So, in these, where these interleukins are coming from, that may be that interleukin 1, 6 and TNF-A is the endogenic pyrogens, and then interleukin 8 and 12 will be the exogenic pyrogens. If the interleukins 1, 6 and TNF-A, it is mainly produced from the hypothalamus of our brain, and then it will be sending the message towards the macrophage. Then the actions will be started, the responses particularly towards that, the microorganism will be started. Then the ILS6 also will produce from the message from our liver. It may be in 2 different phase protein pathways, that may be monos binding actin pathway and C reactive peptine or protein pathway. These 2 pathways may be happen. We will be discussing this part in the next sessions in the following sessions. So, once we completed the phagocytosis action, we also must know that some intelligent bacteria or microbes, it may be having that capacity to escape from these phagocytosis actions. Particularly here I am just showing that the incomplete phagocytosis, that may be the listeria. The 2 bacteria I am taking. First one is the listeria, that is a bacteria that once the first step binding was happened, the second step the engulfment was happened, and engulfment, after the engulfment the cell wall, the temporary cell wall is made. Then after that, before going to the phagosome, the step, the listeria, once the listeria microorganism is entered to our body, what it will do, enter to our macrophage cell, what it will do, it will create that listerolysing, the enzyme known to be the listerolysing, to produce that listerolysing and then cut the temporary cell wall. Once if it is the cut that cell wall, the temporary cell wall, then what it will be actually happen, there is no possibility for ODD or OID. So automatically that cell will be easily affected by that particular microorganism. In the same way, that another one important known bacteria is a mycobacteria. This is also will create that catalyze enzyme and then produce the catalyze enzyme, and then it will be make the same reaction like a listeria to cut that the temporary cell wall. Then once the cut it, then automatically the particular macrophage will be started to infected. So in this way, if this type of bacteria or microorganisms will work in this, then the phagocytosis is not completed until the step 3, 4, 5 is not completed. So it is going to be incomplete phagocytosis. So in this regards we come across with the phagocytosis as well as some example with the incomplete phagocytosis. Next, we will go to that the next important responses of cellular factor, that is the inflammatory responses. We all known to be that the inflammatory responses, we all are come across each and every day with these inflammatory responses. For example, I will show here, this is our skin, so that is going to be our barrier of our immune system, and then that inside the down one is the veins, so which is having that our blood circulation. So if how the inflammation will work or inflammatory responses will happen. So first I will puncture in using any pin or any other particles, then I will puncture our skin in one place. Until that I puncture, there is no blood flow or anything is happened. It means there is no any infectious microorganism are surrounded particular environment. But once you feel pain, you feel blood, blood it is coming from outside from the puncture, then it is known to be that some contamination happened through that unknown microorganism which is present in our cell, our environment. So once if I puncture that particular place in your skin, then automatically what it will do, that microorganisms which is surrounded in that environment, it will be entered to our body. So then immediately our basic immune responding cells like a mast cell, dendritic cell, neutrophil, monocyte and macrophage, these cells will be come to work for that to remove that particular microorganisms. In these, if it is possible to remove that using these 5 different cells, then the first responses is coming from that mast cell. The mast cell, it having the loss of granules inside, that is known to be histomers. These histomers will try to stop that particular microorganisms and then release that histomers towards our blood vessels. So the histomers will cover the blood vessels because the histomers will protect from that microorganism for our particular blood cells or blood vessels. So if after this mast cell performance, it will call for the macrophage. Once the macrophage is coming from that place, then automatically the phagocytosis actions will be start. So using that interleukin 1, 6 and TNF-A. If once the macrophage is also trying to find that complete or eradicate that microorganism from that contamination to our body. But if once macrophage is fine, then the macrophage immediately will call for the dendritic cell. Once the dendritic cells is involved, then automatically what it will happen, we will face the problem like swelling and then the pain on all other things. Once, for example, these dendritic cell once involved in this particular immune response, then it will be collaborate with the complementary system and then produce the 2 different type of toxic materials like the chemotoxics and anaphylaxis. So in this toxic material, it will be produced from dendritic cell. Once it is produced, then it will be try with these toxins to eradicate that particular microorganism which is entered to the particular puncture. Then once even it is not possible by then in the dendritic cell itself, then it will call for the neutrophil and monocytes. Then immediately the actions will start from the monocyte. For that, the action is known to be diaparesis. That is, we commonly known as pain. Once the puncture is happened, immediately we will face the pain. So that known to be the diabetes action. Once the diabetes action is there, then our blood vessels will release that our blood, and then so how much is pressure is needed to eradicate that particular microorganism from our body. Once the diabetes action is over, then it will be immediately call for the other red blood cells to stop these microorganisms enter to our blood vessels. So in this action, once it is over, then we will face the swelling, pain, and some kind of heatness in that place, and redness in that place, like red patches in that place, because of these all five immune responding cells is immediately working this action. This is known to be that inflammation responses once the microorganism enter to our body. So from these, I conclude my session uh, for these cellular factors. Hope we will look into the next session in the complementary system that is known to be the humoral factors.